So thank you very much, Vitrolife, for your invitation, for having me here to talk to all my colleagues. Thank you, David, for the introduction, and thank you for talking after me. It will be a big task, I think. So I was asked to talk about the importance of reproducticity testing of consumables and products in assisted reproductive technologies. It looks very complicated, but we are all being enforced, we are all being controlled now in most countries by legislational bodies. And they will impose on us standards so we can monitor and ensure quality for our work for our patients. And this is true for all the ART laboratories, so the IVF labs, but also for those andrology services where we do sperm analyses or sperm freezing. So we have to work according to rules and guidelines. Now, this has been done to ensure that we give quality to our patients, but there are also guidelines and rules and laws for the producers. So I give you an example here because we are at the Vitrolife workshop. And Vitrolife ensures us, because they have to also comply to these guidelines and laws, that they will give us pharmaceutical quality in ART for their media. What does that mean? Well, that means that they test all their raw materials, they evaluate them individually, and then they will go through a very stringent quality control before they produce the media. They do different tests, the mouse embryo assay, for example, and then they produce the media. Have a talk with them and they will tell you how difficult it is to find really good quality raw materials that pass all these stringent tests. Every lot will undergo, again, then a rigorous quality control testing, including the mouse embryo assay and including endotoxin tests, microbiological tests, and then they are being released to us, the customer. They will give you a certificate, will give you all the details, which we can use for our data and for our database and show to the governments. And they are also obliged to work with a CE label, or if you're in the States, with FDA approval. So this is for the producers. But we as the ART user, we have a big responsibility because we have to choose those reagents, media and consumables that have gone through all those stringent controls. We have to pick out the best quality for the lowest price, of course, because there again stress comes in as a manager. You need to work at the lowest cost possible, but still ensure high quality products that you bring into your lab. So we should ensure that all the above, so the media, the reagents and the consumables, they are tested before we bring them into our lab and use them on our patients, so that they don't have any possible negative effects on the viability or the function of the gametes and the embryos. Now, we had a little bit debate on if it's reprotoxicity or toxicity, but anyway, if there is a toxic agent, there could be a direct or indirect dose-dependent contact, and this could cause a negative influence on the viability and the function of these gametes and embryos. It could be the ovaries, it could be the testes already, but we have to ensure that we avoid this reprotoxicity. Now, how do we see this reprotoxicity? It could be immediate, so all of a sudden you have degeneration of your gametes of embryos. I don't wish that on anybody, but unfortunately I experienced that already 2 times in my career. Once it was when we had a very bad batch of oil and all the oocytes exploded. We could trace it back to the oil. We sent the oil back to the producer, and what they unfortunately did was they sold the same bottles to another lab. So this is like 20 years ago. Luckily, this is not happening anymore. But this is something you can see in your lab. This is really an immediate effect of a toxicity. There could also be an impact on the fertilization process. So you could see lower fertilization rates. It's very difficult to trace back to what's the cause, which product or which consumable will influence your fertilization rate. It could have an impact on the development, so you could have less good quality embryos and a lower implantation rate. But you could have also good implantation rate and high biochemical pregnancy rate or high miscarriage rate. So the manifestation of this reprotoxicity or toxicity can be seen at different stages. There is one publication, and unfortunately, we don't publish enough negative effects. We are afraid to show that we had a problem in the lab, which is actually, it's not a problem, but we should see it as something we can learn from. And this publication, the authors, Ray et al., they saw that a transfer catheter had a negative effect and influenced the toxicity on the embryos, so they had reduced implantation rates and reduced pregnancy rates. Unfortunately, you will only see this after 1 or 2 months, and in the meantime, you are still using the same batch of the catheter, and this is something you can avoid. Well, I will go into the details how we could do that. We need to identify, and I have to stress again, we need to identify those products that we use during the sperm prep, during the storage, during the IUI procedure, during our oocyte collection, embryo culture, embryo freezing, and embryo transfer, and any other procedure that we use, like, for example, embryo biopsy or testicular biopsy. Now, there is a whole range of tests available. I'm just going to briefly go through these tests, and most of you will say, this is out of my league, we cannot do this in the lab, and I will help you go to a test and guide you through this test, which one you can really easily use in your lab. So again, there are different tests that can be used to test influence on female fertility, male fertility, gametes and embryos, and early pregnancy. So let's look briefly at female fertility. There are different tests out there where they culture, it's called a follicle bioassay. So follicles are cultured and matured in vitro, and in this way you can then test different parameters like folliculogenesis, steroidogenesis, and even your your maturation to the M1 or M2 stage of your oocytes. This is the in vitro maturation assay. Of course, this is all done on an animal model. There is also the superovulation study, where you will assess the responsiveness of your ovaries after a product or a medicine has been introduced into this mouse or bovine system. There is also the ESHRE cycle follow-up. So even at these stages, toxicities can go into the system, and this would link into your diet or your lifestyle of the patient, and it can influence the quality of the eggs and embryos and sperm as well. Now, other tests you can do is the mouse in vitro fertilization assay. You explained it in detail. You have to look at the developmental steps of the embryos, look at how fast they're cleaving, the quality, the number of cells, and we can also transfer them back into pseudo-pregnant females, so you can check the implantation sites, you can check if the feti are normal. So all these tests can be done in an animal model. There is also the embryonic stem cell test. These are recent tests, and they're, I think, more feasible. But again, the molecular tests on these embryonic stem cell tests has now been developed. You still are working with an animal model. For male fertility testing, so reprotoxicity, you can culture latex cells, you can look at primary Sertoli cell cultures, and you can do the test that I will go into detail, a sperm analysis test, meaning that you will evaluate the sperm functionality and viability over time. Now, there are different models in different animals. There is the hamster and the mouse sperm motility test. There is the bovine spermatozoa cytotoxicity test, and there is a human sperm survival test. In our system, and I'm talking as well not only for my colleagues in Cardiff, but where I worked before, in the Genk fertility unit, where we did most of this work, we used the human sperm survival test to monitor all batches, all new materials that were introduced into the IVF system. And how do we do that? Is we look at the motility of a prepped semen sample from a patient, or from a patient that came for sperm analysis, and we look after 24 hours and 96 hours after being exposed to a specific product. Now, the details have been published in the paper in 2008. And of course, if you would like to introduce that in your system, you need to submit this to your ethical committee. It needs to be approved. The patients need to give their consent as well. You cannot just use semen that is a leftover of your IVF or your sperm analysis. Patients need to approve that you use it for these tests. And you need to use a normal semen sample. So this means at least 20 million per mil and more than 50% motility, or grade A and B, and more than 5% of normal formed cells. We prep the sperm, we wash it 2 times, then we split it into 2 test tubes. In each test tube we have 1.5 mil, and we should have between 3 to 5 million of good motile spermatozoa. We equilibrate. We always did the equilibration at 36.5 degrees, not 37 degrees. And so each test is then designed to mimic the actual situation where you would use the product. Like gloves, I will go into detail. For gloves, it would be a different test as for a Pasteur pipette, for example. Now, this is a schematic drawing of how the test actually works. And so you have your sperm prep, you split it into 2. One tube has no exposure, the other part of the sample has exposure to a product or a medium or whatever, and then you do your analysis for motility after 24 and 96 hours. And in this way, you can calculate a SPST index, and we just followed the protocol of these authors, Critchlow and Learman. So you just calculate your SPST index, where you have the percentage of motile sperm cells in your control tube and that of the tested sample, and you have your index. If it's more than 0.85, then it is suitable. If it's less, we consider it as being reprotoxic, and we would not use it in the lab. Now, that's all for the theory. In practice, as I said, each test was different, trying to mimic the actual situation. So, for example, for a glove, we would just take a little part of the glove in a sterile way, expose the glove or the media with the sperm cells for 30 minutes to this piece of the glove, remove the piece, equilibrate for 15 minutes, and then put the tubes at room temperature. And for pipettes, it's easier. You flush with sperm suspension, or you leave it 24 hours filled with the sperm suspension. Straws for freezing, we fill them for 30 minutes with the sperm suspension. You can debate on how to do the test, but this is how we worked. Tubes for culture, they were used for culture or semen prep, we had a 24-hour exposure. Petri dishes, we had surface contact for 1 hour. We also did, in the latest phase, we did 24-hour exposure. We tested the specimen container as well as the inside of the lid, and there we had like a 1-hour exposure, thinking that when the patient would produce the sample, it would take about 1 hour before we would take out the sample and put it in a test tube. So in total, we had 2 groups of products and consumables. We had a group of products that were patient-related, that's how I defined it. So these are the products that are used actually by the consultant, the doctors. It's like gloves and needles, it's the IOI catheter, the embryo transfer catheter. It is the tubing that you use during your egg collection. It's the egg collection needle. So these are, as I said, patient-related procedures. And for those products, we did 143 tests. Then we have those products and consumables that are in direct contact with the gametes and the embryos in the lab. And these are those that are related to those procedures. And we did 207 tests. And again, these are all kinds of syringes, denudation pipettes, ICSI pipettes, Petri dishes, multi-dishes, filters, freezing straws, filling tips, and so on. So these were really the products, the materials we used in the lab. Now, what are the results? Surprisingly, we found 10 items when we tested them in the sperm sample. 10 of those items, after 24 hours, the sperm were completely dead, no motility at all. And this was the case for a condom that you use during the egg collection that's pulled over the probe, the egg collection probe, a hysterometer that they sometimes use to open the os, to find the os for during an embryo transfer, and the gloves. Gloves are really, really tricky, and I'll go into detail later why and how this could be. Then we had 4 items that had poor sperm survival after 24 and 96 hours. There was an egg collection needle in there, embryo transfer catheter, hysterometer again, and the inside of the lid of the sperm pot. So that was a big surprise to us as well, because we were completely trusting this material for the safe delivery of the semen to our lab. We had 4 items that only showed reprotoxicity after a longer period, 96 hours. And this was a tubing of the egg collection needle, a petri dish, sterile Pasteur pipettes, and then the cover of a petri dish that we at that time used to do our ICSIs in. So, well, I don't have to give you the conclusion of this outcome. Now, surprisingly, but on the other hand, not surprising at all, 42% of all the items in the patient-related procedures were not okay. They were toxic. Why was this? And when we look at those products used in the lab itself, it's only 10% because we never focused on it. We always were thinking about the lab, everything in the lab needs to be tested. But so this was really a shock for us that we had such a big risk in the patient-related procedures. Surprisingly, there were also batch-to-batch differences. This was minimal, but some products were very good, and all of a sudden one batch would not be good. So this would then again stress and motivate us to keep doing the tests continuously for all the new stuff entering into the lab and in the patient-related procedures. Now I'll just quickly go into the details of which products, just give you a hint, maybe look into your own system. So again, as I said, it's the condom that we use over the probe, and it's very difficult to find a safe one. We have never been able to find one that is not negative in our tests. So we have a considered risk there. We know there is a risk, but until now, and I'm looking now at producers, please make us one that is safe, that has no reprotoxicity. The egg collection needle was very bad, very poor. We talked to the producers. They were surprised as well, and they said, well, it's maybe one needle. We said, no, we repeated the test with another needle that you supplied us. And what they did was they took back this information, went to their manufacturer, their plant, and they rediscussed the materials that they used in the needle, and they came back to us with the same needle but with different materials, and it was a safe needle. So you can use this information and go back to your suppliers because they want to. They are of goodwill. They want to give you safe equipment. As I said, the tubing of the egg collection needle, check that as well. And there are good ones out there as well. But a tricky one is the gloves. So this is a big, big problem. Many of the gloves we tested are very toxic. As you can see, sperms are dead just like within a few hours even. And then you really have to resource and scout the market to find good gloves. There are good gloves, so you can find them. These are then those products that were used in the gamete and embryo-related procedures. And as I said, we had the inside of the sperm pot. We had petri dishes that were not good. And we had one Pasteur pipette that was for us, inferior quality. So we moved away from that pipette, but we gave feedback to the company, and they also acted upon that, but then we used another brand. So why are these products reprotoxic? Why is that? And why is there a batch-to-batch difference? Even if you start off with a good product and one batch is then all of a sudden not good. So one of the reasons could be the endotoxins. And these are being produced during the packaging and handling of the products, of the needle, for example, or the test tubes. So these could be introduced during that procedure, since it's a one one-time error in this handling and packaging procedures. So again, it stresses that you need even to check each batch, even if you have a good outcome in the first place. Now for the gloves, there are all kinds of gloves out there. You have latex or nitrile gloves, and highly likely it's all linked to the chemical composition of these gloves. For example, for nitrile, it's a synthetic polymer, and zinc oxide, sulfur, and other accelerators are being used during the process. And unfortunately, if there is cyanide that could be released during the procedure, so that's really scary if that then enters into your system with the embryos and or oocytes. They could be made from latex. There is a natural latex, there's a synthetic one. And again, the procedure for the chemical production of synthetic latex uses all kinds of chemical stuff, and they could again be toxic for your embryos and gametes. The natural latex, unfortunately, is not a stable composition because it's a natural product. It comes from the trees, and the composition is different each time. So it's very difficult for the producer to control this composition. And it has, therefore, different physical properties. I don't want to scare you, but there is one study out there where they used toxic gloves, which they identified in their tests, and they touched the gloves, they put on the gloves, they touched an embryo transfer catheter for 15 seconds. Then they tested the embryo transfer catheter, and the toxicity was brought onto the embryo transfer catheter. So the message here is, please talk to your doctors and tell them not to touch the embryo catheter, or try to find and resource these good gloves that you can then use. But there is a risk, even one little product in your system could introduce the toxicities on other products. Now, we also have the tubes and the dishes. Why were they negative? Why were they positive? Why did they have a negative influence on the sperm viability? Well, during their production, we have, we're talking a lot about it also in our IVF system now, the phthalates. They are used as plasticizers, and they're a known toxicant. So we are using toxic products. So check your tubes, look at the brand, talk to your colleagues as well, and see what they are using if they have good results. The inside of the sperm pod, the lid, actually is a cover of silicone, and the toxicity came from this silicone that is glued to the inner side of the lid. And again, here you have hydrogen chloride or other products that are used, chemical products, and they could then leave traces on your final product and then hence have a contact with your sperm cells and influence their viability. Now, it's not a new test. I'm not inventing anything here, far from it. I'm just trying to promote it because it's easy. And even at the time of Edwards and Steptoe, they were doing the sperm survival test on their materials, of some of them anyway. It's easy to perform, you have your biological materials in-house, and there are some differences, and we can discuss on this, on the timing of reading your result. Some people say that even after 4 hours, this is a recent publication, 4 hours you can see the outcome, 24 hours. We use the 96 hours as described in the previous publication, so we're happy with it. So you can debate on this. In the US, it's a standardized technique, and it's, they have an external control system, so labs are being sent samples, they have to do the test, and it's included in the control system. So it's nothing new. Lots of people published on this, so again, it's nothing new. You can trace back all these papers and have a look at what their outcomes were. And the advantages are it's a human model, it's not an animal model, it's readily available, as I said already, it's inexpensive, and it's a first-line assay. And it's really important that you do this in your continuous monitoring program. You can cooperate with other ART units. You can phone them and say, hey, I have this result with this batch of this product. Did you find the same, or this one is safe? So give feedback. And this is what we did in Belgium. We talked to one another about qualities. You cooperate with the supplier. You can also say, I'm testing this product from this batch. This is an okay product. I want the whole batch. I make a reservation for the whole batch, so the supplier knows that he will sell you 10,000 catheters, for example, if it's a batch of 10,000. So he's happy because he can sell you the stuff, and you're happy because it's good quality. It's not each time a new batch that you will introduce into your system. Unfortunately, it's a sperm model. So the question is, sperm, does it reflect the effect of these toxicities on eggs and embryos? And of course, hopefully it does, but we don't know. There are some studies on the way that compare these tests on the same product.